Hello, my name is Rachel Jones. I'm a certified industrial hygienist and professor at the University of California, Los Angeles, Fielding School of Public Health. Welcome to Foundational Principles of Health Hazard Evaluation. I will discuss sources of information used to evaluate the health hazards of chemicals. While we can often rely on summaries and analyses by authoritative organizations, such as the International Agency for Research on Cancer, the National Institute for Occupational Safety and Health, or the American Conference of Governmental Industrial Hygienists, industrial hygienists and other occupational health professionals should be familiar with the types of data used to evaluate health hazards, so as to assess relevance of the health hazard to a particular exposure scenario, to precautionarily identify health hazards for emerging chemicals, and also to be able to communicate with toxicologists, epidemiologists, and health professionals that generate data about the health effects of chemicals. The learning objectives for this module are shown here and include find relevant, credible sources of chemical hazard information, including the potential hazards associated with different classes of chemicals and chemical physical properties, differentiate among information source types, and analyze strengths and limitations of sources of information. When characterizing health hazards, all available evidence is used. This can include three major categories of information: in vitro studies, experimental animal studies, and epidemiologic studies. In vitro studies occur in a controlled environment like a test tube or petri dish and involve cells, not whole organisms. Experimental animal studies are in vivo studies and are highly diverse in design. Because experimental animals are not humans, when considering the relevance of in vivo studies to human toxicity, it's important to consider the exposure duration, such as whether the exposure involves one dose or repeated doses, the route of exposure and whether the route is relevant to how humans will take the chemicals into their bodies, and the animal species, which should exhibit similar biological processes to that of humans for the chemical of interest, as well as have a low rate of outcome of interest. Epidemiologic studies, in contrast, count disease outcomes in humans or measure other biological processes related to disease in humans. There are a number of standard epidemiologic study designs, each of which have characteristic strengths and limitations. Developed by Bruce Ames and introduced into use in the 1970s, the Ames test takes advantage of mutant strains of Salmonella that cannot produce histamine, either because of frame shifts or point mutations in the DNA. If the chemical mutates the bacterial DNA, then the DNA will revert to its natural state and the bacteria will be able to grow in the absence of histidine. In this image, there are two test tubes containing Salmonella bacteria, and the chemical is added to one test tube. The bacteria are then put onto a plate that doesn't have very much histidine. The bacteria without the chemical will not be able to grow well on the plate, though a few bacteria may spontaneously mutate and be able to grow. This is the control. If the chemical is a mutagen, the bacteria to which the chemical has been added will experience mutations that allow them to produce histidine and grow in much greater numbers than on the control plate. And you can see this in the figure below because the plate on the top, where the chemical had been added to the bacteria, shows many more spots of bacterial growth than the control plate on the bottom. In vitro testing methods have moved forward because of an interest in moving away from the use of animals for toxicity testing. Tissue culture methods involving human tissues, not just bacteria, have been developed. Organoids are a new sophisticated tissue culture method in which the tissue differentiates into one or more types of cells and can perform some of the basic functions of human organs. For example, organoids of human skin have been developed that can be used for testing irritation to the skin, reducing the need for in vivo studies. The picture here shows a human skin organoid that can grow hair follicles and nerves, which was developed by researchers at the Harvard Stem Cell Institute. Dose response refers to the proportion of subjects that respond in some way to a dose of chemical, whether the subjects are human or animals. When subjects have been tested with multiple different doses, it's possible to create a dose-response curve. The term curve is used because it is common that this response is sigmoidal or S-shaped. The table shows hypothetical data that describe a dose-response relationship. Consider that we performed an experiment where we exposed groups of 20 subjects to 5 different doses of a chemical and had a control group that received no chemical. The control group has a dose of 0 milligrams. The hypothetical results of such an experiment are shown where no subjects exposed to 0 or 1 milligram of the chemical responded to the outcome of interest. Among the 20 subjects exposed to 2, 3, or 5 milligrams of the chemical, 3, 8, 16, and 18 subjects responded, or 15%, 40%, 80%, and 90%, respectively. One thing to notice in these data is that there is a dose where there is no response. This event could suggest that there is a threshold that defines a dose below which there is no response observed. That is, there is no toxic effect at this dose. The figure on the left shows the same data from the table, plotted, where the x-axis has the dose in milligrams, 0, 1, 2, 3, 4, and 5, and on the y-axis it shows the frequency of the response, or the percent of the exposed subjects that responded. And you can see here that we have the beginnings of an S shape. This is a dose-response curve. Let's say we used the same study design but test a different chemical, or perhaps even test the same chemical on a different species. Then we'll get a new set of results. These hypothetical results are shown in the table. For the dose of 1 milligram, there were 2 of the 20 exposed subjects, or 10%, that exhibited the response of interest. And for the doses of 2, 3, 4, and 5 milligrams, 20, 70, 90%, and 90% of subjects responded respectively. In the figure on the left, these data are plotted in red using triangles, and the black lines and circles are the same data that we saw in the previous slide. With these new data, we do see that there is a response at each of the non-zero doses that were tested with the subjects. There may still be a threshold, but it would be at a dose that is lower than the lowest dose administered in this study. And one would need to repeat the study and expose the subjects to a series of lower doses to determine if it still existed. There are some health outcomes, such as cancer, for which one would never expect a threshold. An important observation from dose-response curves is the steepness of the slope. When the slope is steep, it means that small changes in dose can result in large changes in the response. This is a problem for chemical safety because a small change in the exposure can have a significant impact on workers' health. Looking at the figure, we can see that the dose-response curve in red to the left rises more steeply than the dose-response curve shown in black. One metric that is often reported in safety data sheets and tested for chemicals is the median lethal dose, also known as the LD50. The median lethal dose describes the one-time dose that results in death of 50% of the exposed animals. Typically, the LD50 is tested by having groups of subjects exposed to graded doses of a subject and observing the proportion that die, just like in the data tables shown in the prior slides. An algorithm is then used to calculate the LD50, but you can estimate the LD50 from the dose-response curve. In the figure, a blue dashed horizontal line is drawn at the frequency of 0.5 or 50%. And when you read across the blue line, you see where it intercepts the dose-response curve in red and the dose-response curve in black. For the line in red, the 50% value is at the dose of 2.6 milligrams. For the line in black, the 50% value is at a dose of 3.25 milligrams. The chemical whose dose-response curve is shown in red is more acutely lethal than the chemical whose dose-response curve is shown in black. That is, it is more potent. The LD50 test is performed with varied animal species, but most commonly rats, mice, and guinea pigs. The LD50 test is also performed with various exposure routes, including ingestion and injection. When the exposure is through inhalation, however, the term median lethal concentration, or LC50, is used instead of the median lethal dose, or LD50. Subchronic and chronic studies involve repeated dosing of subjects. Subchronic studies are usually conducted for about 3 months, but the duration can vary from 14 days to 4 to 6 months. Subchronic studies are used to study the impact of the chemical on biological systems, including precursors of disease, or on non-cancer outcomes, such as neurological effects. Chronic studies are typically conducted over a 2-year period and used to study cancer outcomes. Reproductive and developmental toxicities can span multiple generations. These studies may expose mothers or fathers at certain periods pre-conception or during fetal development, and examine pregnancy outcomes or even disease development in the next generation. Sometimes in these studies, doses will continue into the next generation. Overall, there are well-defined protocols for subchronic and chronic studies in animals, designed specifically to assess different types of outcomes. These studies always include a control group in which there are no exposures to the chemicals. It is common for subjects in these studies to be exposed to the chemical on a schedule, such as every day for 5 days of a week over the study duration. Thus, these are repeated dose studies. Subchronic and chronic studies in animals seek to identify different effect levels. 4 key effect levels are shown here. The NOEL, N-O-E-L, or the NOAL, N-O-A-E-L, are the lowest doses for which no effect was observed. That is, it could not be distinguished from the control group. The LOEL, L-O-E-L, or LOAL, L-O-A-E-L, are the lowest doses for which an effect was observed and could be distinguished from the control group. Not all observed effects are considered adverse. An adverse effect is one that causes an alteration of morphology, function, capacity, growth, development, or lifespan. An example of a non-adverse effect might be transient inflammation. The classification of effects as adverse or non-adverse is a significant debate in the toxicological literature. Epidemiologic studies, as I mentioned previously, are studies that involved counting disease outcomes in humans. There are a number of common study designs. Case reports or case series is a type of study design that is generally considered to lack rigor, but may highlight unexpected health outcomes that could be associated with occupational exposures. Such a study might describe the development of a disease among 2 people who perform the same job, where this type of disease may not have been identified among workers in this occupation before. Case control studies compare 2 groups of people, in which one group has the disease and the second group doesn't have the disease. This type of study seeks to determine if one group was more likely to have been exposed to a substance than the other group. For example, the epidemiologist identifies all people in a community that have a disease and finds people of similar age and sex who don't have the disease. Then the epidemiologist looks to see if the proportion of people who have exposure to a chemical is the same among the people with and without the disease. If the exposure to the chemical is more common among the people with the disease than the people without the disease, then we may conclude the chemical is associated with the disease. A cohort study compares the health outcome of groups of individuals who differ by one characteristic, such as exposure to a chemical. For example, a cohort of workers differ in the magnitude of their exposure to a chemical, even if they all worked in the same workplace or the same industry. And the epidemiologist looks to see if the proportion of workers with the disease is the same or different among workers with no, low, or high levels of exposure. When this study follows people over time, it is referred to as a longitudinal study. Cross-sectional studies describe the disease outcome that occurs among people at one point in time. Such a study, for example, may go to a workplace and survey workers present at the workplace to determine how many people have a disease today. This type of study may also collect information about chemical exposures and describe the distribution of disease among people at this time today who have different levels of exposure. Ecological studies do not have data about individuals, but collect data about groups of people only. For example, rather than identifying individuals with a disease, the outcome may be the rate of a disease in a group, such as at a workplace or in a county. Ecological studies are not very common in occupational health research. In assessing causal association between exposure to a chemical and a disease, longitudinal cohort studies are considered to be the strongest design because the epidemiologist can track the temporality of the exposure and the disease outcome. That includes the latency between exposure and disease onset. However, owing to the challenges of implementing epidemiologic studies, health hazard assessments consider all available evidence, including other types of epidemiologic study designs that may provide evidence of association between an exposure and a disease, but not evidence of causation between the exposure and the disease.